Summary of the effects of tuberculosis on the endocrine system. In addition to its well known effects on the respiratory system, tuberculosis, TB, also affects the endocrine system in a variety of ways. TB patients exhibit abnormal levels of multiple hormones, including increased levels of interleukin 6, IL 6, cortisol, growth hormone, GH, estradiol, prolactin, thyroid hormones, T3 and T4, decreased levels of dehydroepiandrosterone, DHEA, testosterone. These changes are not typical of what is seen in patients experiencing chronic stress, suggesting that they are not solely a result of the stress of the disease. Instead, they are likely caused at least in part by cytokines released during the immune response to Mycobacterium tuberculosis. One study found that supernatants from M. tuberculosis antigen-stimulated peripheral blood mononuclear cells, PBMCs, from TB patients inhibited DHA production by a human adrenal cell line, supporting the idea that immune cell products can affect adrenal steroid production. The endocrine changes seen in TB patients may contribute to the pathogenesis of the disease. For example, decreased levels of testosterone and DHEA could contribute to the progressive debilitation and other disabling symptoms that characterize tuberculosis. Increased T3 and T4 levels, which promote a hypercatabolic state, could further worsen this situation. It has also been proposed that the endocrine dysregulation seen in TB may negatively impact thymic function. The thymus is a central lymphoid organ critical for T-cell development, and disruptions to this process could hinder the body's ability to control TB infection. More research is needed to understand the full extent of TB's impact on the endocrine system, particularly how these hormonal changes affect disease progression and patient outcomes. Future research to dissect the effect of TB on the endocrine system. Large-scale studies are needed to better understand the complex interactions between the endocrine and immune systems during TB, especially the role of innate immunity. The sources suggest future research should focus on evaluating the potential of hormones and cytokines as biomarkers for TB treatment response. For example, DHEA, amylin, and T4 could be useful for predicting unfavorable treatment outcomes. Further investigating the impact of adrenal steroids and leptin on thymic function during TB. This could involve studying thymic affectation during active disease and the impact on the immunological status of patients, particularly in the context of progressive disease, multidrug-resistant TB, or HIV co-infection. Examining the role of individual cytokines in mediating